We're going to take a look at energy storage and use and the metabolism of carbohydrates. So we're going to start by looking at how our nutrients in our diet are going to be transported in the blood. So carbohydrates are going to be broken down into the monosaccharides such as glucose. And glucose is hydrophilic, meaning it likes to mix with the water in the blood. So it's really easy to transport just as glucose. And then proteins that we eat in our diet, they're going to be broken down into amino acids and transported just as amino acids in the blood, because amino acids are also hydrophilic and they mix really well with the water in the blood. But lipids are very different because they're hydrophobic, so they don't like to mix with the water in the blood. So to get them to mix better, we're going to package them with some proteins and make these lipoproteins in order to transport the lipids in the blood. And here's a picture of a lipoprotein over here. And you can see that we've got all of these lipids here. There's some phospholipids, some triglycerides, some cholesterol, and then in that we've also put some of these purple proteins. And the proteins are hydrophilic, and so that'll make this lipoprotein mix better with the water in the blood, and that's how we're going to transport the lipids in the blood as these lipoproteins. So with the nutrients that you eat in your diet, the carbohydrates, fats, and proteins, there's three possible things that we can do with them. So one is that we can break them down and use them for energy. So we can use them for making ATP, and then we can use that ATP to do work in the cells. The second thing we could do with the nutrients from our diet is we can use them to make other things that we need in our cells. So maybe we could use them to make proteins that we need in the cells, or maybe we need to make some other types of carbohydrates or nucleic acids. So there's all kinds of things that we can make in the cells. And then if we have some extra nutrients, we can store them for later, because when we evolved, there weren't grocery stores, so we never knew when our next meal was coming. So we would just store any extra food that we ate as these energy storage molecules. So we've got glycogen, and that's the storage form of glucose. So glycogen is a polysaccharide. It's just a bunch of glucoses that are joined together. And it's going to be stored in two places in the body. So it'll be stored in the liver and also in skeletal muscle cells. And then triglycerides, also known as fat, that's going to be stored in the adipose. And then we also have proteins. So they're going to be stored in the skeletal muscle, but that's going to be kind of a last resort. So you're only going to be starting to break down those proteins to use as an energy source from your skeletal muscle when you're starving. So you'll go through your triglycerides and your glycogen stores first, and once those are gone, then you'll start breaking down the proteins in your muscle. So those are kind of a last resort. So this is a really nice table from your book. This is from chapter 21, table 21.1, and it shows the three different nutrients from the diet and how they're absorbed across the GI tract, and then how they're transported in the blood, how they're stored in the body, where they're stored, and then the percentage of the total energy that's stored in the body. So most of the energy that's stored in the body is going to be as fat, as triglycerides, because we do unfortunately have an unlimited store of fat that we can have, because you can keep expanding those fat cells. And then we do have quite a bit of protein in our muscle cells, but that's kind of a last resort. We're not going to touch that unless we're starving. The glycogen stores in the liver and skeletal muscle, those are actually pretty small. We don't have a whole lot of glycogen. So this picture from chapter 21 is also really important to understand. So I definitely would know this. This picture could show up on the exam as well. So make sure you understand what's going on with each of the three nutrients. So first we've got glucose. So we've got glucose that's been absorbed from the GI tract being transported in the blood. And to get the glucose from the blood into the body cells, glucose is hydrophilic. So it's not going to just diffuse through the cell membrane. We need a transport protein. So that's what this purple thing is. So we're actually going to use a carrier protein, and we're going to transport it into the body cells using facilitated diffusion. And once the glucose is transported into the cell, we're going to phosphorylate it. And the reason we do that is once you stick a phosphate group onto the glucose, it changes its shape, and then it can't get transported back out again. So that's good because glucose is valuable and you don't want to spit it back out again. So once the glucose is inside the cell, we have three possible things we could do with it. So one is we could break the glucose down into carbon dioxide and generate ATP and use that to do work in the cell. We could certainly take the glucose and make other things with it. And then we could also take the extra glucose that we have and store it for later as glycogen. And then if we're running low on nutrients, maybe it's been a couple hours since we've eaten, we can certainly break down the glycogen and chop off the glucoses. And then in the liver, especially and a little bit in the kidneys, we actually can remove the phosphate group and then release the glucose back into the blood and raise our blood sugar when it's getting too low. So that's a good thing to be able to do. Okay, next we have our proteins. So they're going to be transported in the blood as amino acids. Amino acids are hydrophilic, so they cannot diffuse through the cell membrane. So we're going to have a transport protein transporting them into the cell. And once they're into the cell, we can certainly break down the amino acids and make ATP with them, use that to do work. And when you break down the amino acid, the amino group on the amino acid, we can't do anything with that. So we just chop off that amino group and we send that over to the liver. The liver converts the amino group into urea, which is a waste product that gets filtered out of the blood by the kidneys and gets excreted into the urine, and then it's gone. If we have extra amino acids, or maybe we just need to make some proteins, we can make some of those proteins. And then if you are starving, we can break down those proteins and release the amino acids and then make ATP with that. And then our last nutrient, the lipids. So remember that the lipids are hydrophobic, so they don't mix well with the water in the blood, so we're going to package them as these lipoproteins. And as they're traveling through the blood, there are some enzymes located on the capillary walls. These are called lipoprotein lipases. We'll talk about these in another lecture. And the lipoprotein lipases will grab the lipoproteins as they go by and rip off chunks from the lipoproteins. And we'll take the triglycerides and break them down into monoglycerides and fatty acids. And then some of these will go to the liver for further processing, and then these fatty acids, being hydrophobic, can just diffuse right out of the bloodstream, cross the cell membrane, and go right into the body cells. So we don't need a transport protein for these fatty acids because they're hydrophobic. And once inside the cell, the fatty acids can be broken down into carbon dioxide, and we can get ATP from that to do work. If we have some extra fatty acids, we can certainly make some triglycerides if we're in our fat cells, our adipocytes, and store that for later. And if it's been a while since we've eaten, we can certainly take those triglyceride stores in the fat cells and break those down into the fatty acids and the glycerol. release that into the bloodstream, and then those can go to the liver and other body cells and then be used by other cells for energy. All right, so let's focus on carbohydrate uptake and metabolism first. So if we take a look at our picture over here, how does glucose enter the cells? So remember that glucose is hydrophilic, mixes very well with the water in the blood, but that means that it can't cross cell membranes. So we're going to need a transport protein, and we're actually going to use a carrier protein. And so we're going to transport the glucose across the cell membrane using facilitated diffusion. So that's how it's going to enter the cells. So we're going to use facilitated diffusion. And there's a hormone that will help glucose enter a lot of the body cells. This hormone will tell the body cells that, hey, you just ate and there's a lot of glucose out there, so put out your carrier proteins in your cell membrane and start taking that glucose in. And you probably guessed, the hormone is insulin. And insulin is secreted by your pancreas. Okay, we do have some enzymes that are involved with glucose metabolism as well. So once we transport the glucose inside the body cell, we're going to phosphorylate it. Okay, so we're going to stick a phosphate group onto it, and that way we can't transport it back out because we want to keep it inside because it's pretty valuable and we don't want to spit it back out. So by putting that phosphate group onto the glucose, it changes shape and then we can't transport it back out. So we're going to use a kinase enzyme to phosphorylate the glucose, because remember, kinase enzymes phosphorylate things. So that's called a glucokinase enzyme. So just remember kinase enzyme, if you see that, it phosphorylates things. And then if you're, maybe a couple hours, it's been since you've eaten, your blood glucose is getting low, and you want to release some glucose back into the blood to raise your blood sugar, the liver and a little bit the kidneys can do this. They have an enzyme, phosphatase, that will dephosphorylate that glucose, and that'll allow you to transport it back out into the blood. Okay. All right, so once the glucose is into the cell, there's a couple things we can do with it. One is that the glucose can start down the first step of aerobic respiration and enter into glycolysis. And what we're going to be ending up producing at the end of aerobic respiration is ATP. So that'll be our answer there. And we'll use that ATP to do work in the cells. So maybe we're doing chemical work, doing anabolic reactions, maybe we're doing mechanical work, maybe contracting muscles, or maybe we're doing transport work. Maybe we're doing some kind of active transport. Okay, and then another pathway we could take, if we have extra glucose, we could store it for later and join the glucoses together to make glycogen. And the term we use to make glycogen is called glycogenesis. And genesis just means to form or produce, and glyco is talking about glycogen. And remember that the two places in the body where glycogen is going to be stored is in the liver and skeletal muscle. So that would be good to remember. And then if it's been a couple hours since you've eaten and blood glucose is running low, you can break down the glycogen and chop the glucoses off. And breaking down the glycogen, that's called glycogenolysis. Because remember, lysis means to break apart or split, and we're breaking apart the glycogen. So a lot of that will happen in the liver and a little bit in the kidneys. and they have that phosphatase enzyme to dephosphorylate the glucose, and then you can release the glucose into the blood, and that'll raise the blood glucose levels back up to normal. Okay, and then the other thing we can do with the glucose is we can certainly make other things with it in the cell if we need to. Okay, so just looking at what's going on in the liver and kidney with these enzymes. So here we've got the glucose, and when it gets transported into the cell, it'll get phosphorylated, and we've got that kinase enzyme that phosphorylates the glucose. And then if we're running low on glucose, it's been a couple hours since we've eaten, the liver and also the kidney have a phosphatase enzyme that will dephosphorylate the glucose, and that allows the liver and kidney to move the glucose back out into the blood, and then that'll raise the blood glucose. Okay, so let's just draw this out. So here we've got some glucose, and we're going to have it come into the cell here. And when it comes in, we're going to stick a phosphate group on it. And so what's going to do that is a kinase enzyme. So if you just remember kinase, that phosphorylates things. And then the liver and also a little bit the kidney can take the glucose and dephosphorylate it. It can chop off that phosphate group, remove it, and it does that by using a phosphatase enzyme. Because remember, phosphatase enzymes dephosphorylate things. Okay, so, and then that will release the glucose back into the blood, and that'll raise the blood sugar because it's gotten too low because you haven't eaten for a while. Okay, so the liver will do that, a little bit of the kidney. Skeletal muscle, though, which also stores glycogen, does not have that phosphatase enzyme, so it can't release glucose into the blood. So it can't do that, but the liver can. Okay, another G word, we've got gluconeogenesis. And if you take a look at this word and just break it down, we've got gluco referring to glucose, neo is new, and gen means to produce. So gluconeogenesis literally means you're producing new glucose, and we're making new glucose from non-carbohydrate things. Okay? All right, so what are we doing? All right, so we're going to be taking glycerol, which is part of triglycerides. So that comes from triglycerides or fat. Oops. And so we can actually take triglycerides and make glucose from that. This is really cool. And so that's a really good thing to be able to do. If you haven't eaten for a while, you can take your fat and make glucose from it, and that'll raise your blood sugar. You can also take lactate, which is also known as lactic acid, and that comes from fermentation, right? And you can turn that into glucose. And then you can take amino acids, which come from proteins, and you can make glucose from that. So there's a lot of different things that are not carbohydrates that you can make glucose from. All right, and so this gluconeogenesis is going to occur mostly in the liver and a little bit in the kidneys, and it's going to occur in what we call the post-absorptive state. And what the post-absorptive state is this has been at least 3 hours since you've eaten, and your nutrients are starting to run low in your blood. Okay, and so your liver senses that and says, hey, our blood glucose is getting low. We need to raise our blood glucose. So let's start making some new glucose from things that aren't carbohydrates so it can tap into your fat stores, lactic acid, into the amino acids, and make new glucose from that. And then that'll raise the blood glucose. All right, so here's a crazy picture from chapter 3. Don't get alarmed at this. It's just showing you that you can take amino acids and work your way over and make glucose from the amino acids. You can take glycerol, which is part of the triglyceride or fat, and make glucose from that. And then you can also take lactic acid from fermentation and work your way over and make glucose from that. So just showing you how you can take these non-carbohydrate things and make new glucose from that. So that's gluconeogenesis. All right, so let's summarize this. We've got a lot of G words running around, and so you need to keep these straight for the exam because you know I'm going to put this on the exam. All right, so let's fill this in and make sure that we've got all of these straight. So if you're going from glucose and breaking it down to make the ATP over here on the right, that first step of aerobic respiration, that is glycolysis. Okay. All right. And then if you have some extra glucose and you want to store it for later, you can join the glucoses together to make glycogen. So going from the glucose to make the glycogen, that's called glycogenesis. Remember, glyco is referring to glycogen, and then genesis means to form or produce. And then if it's been a while since you've eaten and you need to release some glucose into the blood, you can take your glycogen stores, like in your liver, and you can break those down into the glucose. So breaking the glycogen down into the glucose, that's called glycogenolysis. So you have glycogen, and then lysis is to split or break apart. Okay, and this last one on the bottom, we've got some non-carbohydrate things. We've got glycerol coming from your triglycerides or fats. You've got lactate or lactic acid from fermentation, and you've got amino acids from proteins, and we can take these things that are not carbohydrates and we can make glucose from that. Okay, and that is called gluconeogenesis. So we're making new glucose. Okay, and that'll happen in the post-absorptive state. It's been a couple hours since you've eaten, your blood glucose is low, and your liver will do this. Okay, and it'll make this new glucose and release that into the blood, and then that'll raise your blood glucose.